This is an overview of the male reproductive system. We'll begin by looking at the embryonic development of the reproductive system so that we can compare and contrast the male and the female system. At an early stage of gestational development, about weeks 5 to 6, the reproductive organs are not yet differentiated. There is an undifferentiated genital ridge that grows associated with the early stages of kidney development. Parallel to the genital ridge, there are two ducts, a mesonephric duct and a paramesonephric duct. One of these ducts will develop in the male and the other will develop in the female. There is also, in terms of the urinary and reproductive tracts, a single opening called the cloaca. In the male, the presence of testosterone at an early stage of development spurs the deterioration of the paramesonephric ducts and persistence and development of the mesonephric ducts. The genital ridge differentiates into testes, and the mesonephric ducts grow ductules into the testes. At the urinary tract, there is now a urogenital sinus forming the urethra, and the mesonephric ducts grow into and connect with the urogenital sinus. Notice that the seminal vesicles are also developing. By the time of birth, the prostate gland has appeared, the bulbourethral glands have established, the urethra runs its entire length, and the testes have at least begun their descent into the scrotum. In the female, by contrast, the paramesonephric ducts persist and the mesonephric ducts deteriorate. The genital ridges on each side differentiate into ovaries. The paramesonephric ducts merge to form the uterus, and the uterus connects to the urogenital sinus, which will further develop into the vagina and the urethra. At birth, the paramesonephric ducts have fully established themselves as uterine tubes. The infundibulum is approaching the ovary, which it will later cover. The uterus is fairly fully developed, as is the vagina. The upper portion of the vagina actually grows off of the tissue that has developed into the uterus, whereas the lower portion of the vagina develops from the urogenital sinus. Development of external genitalia parallels the development of the internal reproductive organs. Again, at about week 6, we are sexually undifferentiated. A genital tubercle sits anteriorly past the urogenital folds on either side. Labioscrotal swellings surround the urogenital folds. In a male, the two urogenital folds will fuse into the body of the penis, and the genital tubercle develops into the glands of the penis. In the female, the urogenital folds persist as two separate structures, becoming the labia minora, and the labioscrotal swelling become the labia majora. The genital tubercle develops into the clitoris. Once fully differentiated, the scrotum, which developed from the labioscrotal swelling, encloses the testes in the male, and in the female, the vaginal orifice sits in between the two labia minora. Looking at a sagittal view of male reproductive anatomy, we can see some structures that have been sliced through at the mid-sagittal point. Other structures are shown parasagittally. In the mid-sagittal view, we can see the section through the penis, the glands of the penis covered by the prepuce or foreskin. You can see the erectile tissue within the penis, the urogenital diaphragm, which contains the very small bulbourethral gland. This bulbourethral gland is a parasagittal structure. Its duct empties into the penile or spongy urethra. The prostate gland, which is situated superior to the urogenital diaphragm. The main passageway through the prostate gland is the urethra, but secondarily the ejaculatory duct from the left and right seminal vesicles pass through the prostate to empty into the urethra. Seminal vesicles, again, parasagittal structures. There's a left and a right, produce about 60% of the semen. Adjacent to them, the ampulla, which is the widest portion of the ductus deferens. Tracing the ductus deferens backwards to its origin within the scrotal sac at the base of the testis. Sitting beside it, covering the testis, is the epididymis. So the pathway for sperm is from the testis to the epididymis, to the ductus deferens, through the spermatic cord, emptying at the ampulla into the ejaculatory duct, passing into the prostatic urethra, through the very short section called the membranous urethra, to the penile or spongy urethra. In a frontal view, you can see that the scrotal sac, the scrotum itself, has two lobes. Each lobe contains a testis, an epididymis, and a spermatic cord. The spermatic cord connects the scrotal sac to the abdominal pelvic cavity. It contains a testicular artery, which is surrounded by a network of veins called the pampiniform plexus, which are the drainage of the testicular vein. So here sit testicular artery and vein. The opening through which the structures of the spermatic cord will pass is called the inguinal canal, and it is reinforced externally by an inguinal ring of connective tissue. The spermatic cord itself has a covering over it, the spermatic fascia. The scrotal sac has a layer of muscle called the dartos muscle, which can contract to bring the testes closer to the pelvic floor. Within the spermatic cord, there is also a cremaster muscle, which can contract to shorten and pull the testis closer to the pelvic floor. Here in a posterior view, you can see this paired seminal vesicles on the posterior surface of the urinary bladder. Testis epididymis, ductus deferens ending at the ampulla, and emptying its contents into the ejaculatory ducts, left and right, within the substance of the prostate gland. Prostatic urethra, membranous urethra, penile urethra or spongy urethra, bulbourethral glands situated within the urogenital diaphragm. We can also in this view see the separate bodies of erectile tissue in the penis. There are two corpora cavernosa and a single corpus spongiosum. The corpus spongiosum contains the urethra. The crus is the upward extension of the corpus cavernosum on each side, embedded in the pelvic floor. The glans is the expanded distal end of the penis, and again, ordinarily covered with a prepuce or foreskin. Here in cross-sectional view, you can see the right and left corpora cavernosa. Each corpus cavernosum has in its deep interior a central artery. The cavernous spaces or venous spaces within each body of erectile tissue can become engorged with blood, creating an erection. The corpus spongiosum contains the urethra. Here we see the internal structure of the testis. The testis is subdivided into lobules by connective tissue septa. Within each lobule there is 1 to 4 separate seminiferous tubules. Each seminiferous tubule is highly folded and packed into the lobule. Seminiferous tubules are the site of sperm production. Sperm, once it's produced, will move through the seminiferous tubule and into this series of ducts. This network right here is called the rete testis, and the ducts that leave the rete testis enter the efferent ductules, which go into the head of the epididymis. the head of the epididymis has multiple ductules, but by the time you get into the body of the epididymis, there is a single duct. The tail of the epididymis makes a hairpin turn, and the duct of the epididymis is continuous with the ductus deferens. The connective tissue wrappings around the testis and epididymis include the tunica albuginea, which is the outer covering, the fibrous capsule of the testis itself, and a double-walled tunica vaginalis, which is the extension of the serous membrane that accompanied the testis when it descended from the abdominopelvic cavity into the scrotum. You can also see the elements of the spermatic cord here. Here's a cross-sectional view of a single seminiferous tubule. The chromosomes are stained dark in this view, as are the heads of the sperm cells. So here are the heads of sperm. The tails of the sperm, light blue, fill the lumen. The heads of the sperm are embedded within the Sertoli cells, also called sustentacular cells. Sustentacular cells make up the wall of the seminiferous tubule. All of the cells that contribute to sperm formation are embedded within the sustentacular cells. The germ cells themselves include the spermatogonia, and then the different stages of development, including the spermatocyte stage, as we'll see. Interstitial cells surround the seminiferous tubules, and they are the site of production of male hormones, primarily testosterone. Sperm production begins on the peripheral edge. Spermatogonia divide by mitosis to replenish their population. So as a cell divides into two, one of the two daughter cells will move inward, enlarge into a primary spermatocyte, and begin meiosis. Hormonal regulation of male reproductive function begins in the hypothalamus with GnRH, gonadotropin-releasing hormone, which is chronically secreted in males. It travels to the anterior pituitary through the portal system between the hypothalamus and the pituitary. Primary hormones secreted by the anterior pituitary in males are FSH and LH. They have very different functions. LH stimulates interstitial cells to produce and secrete testosterone. FSH stimulates the sustentacular cells to secrete the receptor for testosterone, which is the androgen binding protein, ABP. Androgen binding protein holds testosterone within the cells of the testes. Testosterone stimulates spermatogenesis, but also provides negative feedback to regulate GnRH production. Sperm count is specifically controlled through negative feedback through a hormone called inhibin, which also provides negative feedback to GnRH. In addition to stimulating spermatogenesis, testosterone also has other effects on the body, stimulates libido and development of secondary sexual characteristics in males. Spermatogenesis is the product of meiosis. Meiosis is nuclear division for the formation of gametes. The steps of meiosis parallel the steps of mitosis. However, in meiosis, we're going to go through these steps twice. So we have two phases of meiosis. We simply call them meiosis 1 and meiosis 2. The first step, not shown in this image, is the replication of the DNA, giving us chromosomes that have already doubled. In prophase of meiosis 1, the homologous maternal and paternal chromosomes will pair up with each other, and very often they will swap genetic material. This pairing up is called synapsis. Swapping genetic material is called crossing over. Synapsis and crossing over increases the genetic variability within the gametes. At metaphase, anaphase, and telophase, we're going to take the chromosomes and separate them. We have what we call independent assortment of chromosomes. That is, there is no requirement that all the paternal chromosomes stay together and all the maternal chromosomes stay together. They mix at random. So by the time you finish with telophase 1, each of the two daughter cells that is developing has a random mix of paternal and maternal chromosomes. As we go immediately into meiosis part 2, chromosomes realign themselves on the equator at metaphase. At anaphase, we are now separating the two halves or sister chromatids of each of the chromosomes so that the resulting daughter cells have only one set of chromosomes. This condition is called haploid. In humans, the haploid number is 23. Because of crossing over, no two gametes are identical. So the result of meiosis is 4 non-identical haploid gametes. As we watch meiosis occurring within the testis, in the seminiferous tubule, we begin with the spermatogonium, which divides not by meiosis, but by mitosis. As it divides, one of the two daughter cells will simply stay right where it is. The other one will literally migrate, enlarge, and become a primary spermatocyte. The primary spermatocyte will undergo meiosis 1. Remember that means that the two daughter cells that are the result of that have only half the number of homologous chromosomes. This cell is called a secondary spermatocyte. It goes through meiosis 2, producing non-identical spermatids as the chromatids separate. Spermatids mature into sperm cells by a process called spermiogenesis. In spermiogenesis, the spermatid grows a flagellum, which is the tail of the sperm, removes excess cytoplasm, and has a very elaborate spiral mitochondrion that powers the swimming of the flagellum. The head contains the nucleus and a specialized lysosome called the acrosome.